And this image just reiterates what I was talking about where the spikes are located versus a naked virus and envelope virus. So we have two different types of virus here. We have a naked virus, which is just going to be the virus's genetic material in the nucleic acid core, which we can see here, a protective layer known as the capsid. And then since this is a naked virus, the spikes are going to be present out here on the capsid. And then with an envelope virus, you have the nucleic acid core with the genetic material. You have the capsid that surrounds the nucleic acid core for protection. And then since this is an envelope virus, it has the additional layer, the additional envelope layer here surrounding the capsid. And then on the envelope are the spikes that are used for attachment to additional host cells. So we're going to talk about each viral structure separately to go through kind of function and purpose. So all viruses have a capsid. Capsid is essential for all viruses. Its primary purpose is to surround the nucleic acid core, so surround the genetic material of the virus, and essentially protect that genetic material. So when we talk about capsids, they're mandatory for all viruses. Capsids are made up of proteins called capsomeres, which is easier to see in this first image. So this purple layer surrounding the genetic material is the capsid. And then each of these individual circular structures here are capsomeres. And these capsomeres will come together like puzzle pieces around the genetic material to make up the capsid that will protect that nucleic acid core. So the main purpose of the capsid and the capsomeres that make up the capsid is for protection of the virus's genetic material. Depending on how the capsomeres link around the genetic material to form the capsid, that can dictate what shape the virus is going to be. So viruses can have multiple shapes, not nearly as many as what we've talked about with bacteria, but one shape that they can have is icosahedral, which is what we're seeing here. Icosahedral means many triangular faces. So viruses that have an icosahedral shaped capsid are going to pretty much look like diamonds with the multiple triangular faces. And then some viral capsids are helical in shape. So we're kind of seeing that distribution here where they have this helical arrangement as far as the capsid. And then some viruses are called complex in shape. They're called complex because they have multiple shapes that make up one single virus. The only substantial virus that is considered complex in shape is going to be bacteriophages, which are viruses that can infect bacteria. They actually have icosahedral heads, which is what we see right here, and then inside of their icosahedral head there would be genetic material in here. But then their neck region is actually more of a helical shape. So we already have kind of those two different shapes in one virus here. And then they actually have these little tail fibers that essentially serve to attach them to different bacterial cells for infection. But because they have multiple shapes making up one viral unit, they're considered complex in their shape. While all viruses have a capsid that's meant to protect the virus and or viral genetic material and give the virus shape, the viral envelope is an optional layer that only envelope viruses would have. So viruses that lack an envelope would then be referred to as naked viruses. So the viral envelope, if a virus has it, is going to set right outside the capsid. So if we have the nucleic acid core here in the red, which is the genetic material, we would have the capsid surrounding that here for protection, and then this surrounding the capsid would be considered the envelope. And the envelope is made up of the same thing that our cell membranes are made up of, being things like lipids, carbohydrates, and proteins. And that's because the envelope originated as a cell membrane from a host cell. So when viruses replicate inside of a host cell and then eventually leave that host cell so they can go on and infect another host cell, they actually take a piece of the cell membrane with them in a process known as budding. And that piece of the cell membrane that they take with them during the budding process is what will actually serve as their envelope. So anytime we see an envelope, that envelope actually originated from a host cell membrane at one point. But the envelope does provide an additional layer of protection for the virus. This, keep in mind, only envelope viruses would have this envelope. And then on the envelope, we would have these spikes or glycoprotein spikes. These spikes serve to attach the virus to host cells when the virus is trying to penetrate a host cell. Just keep in mind, with naked viruses, since they don't have an envelope, obviously those spikes would need to be on the capsid to attach the virus to a host cell. So with the nucleic acid core, it can get very complex depending on what type of virus we're talking about, because there are some viruses that are DNA viruses, so they're going to have DNA in their nucleic acid core. And then there are other viruses that are RNA viruses, they're going to have RNA in their nucleic acid core. But then there's different types of DNA and different types of RNA depending on what type of virus we're talking about. For the context of this course, all you need to know is that in the nucleic acid core is where we find the virus's genetic material, and that is either going to be DNA or RNA, but never both. And with viruses, they have their genetic material in the form of DNA or RNA, but they don't have an abundance of it. So to kind of put this into context, if we look at our DNA in our cells and we stretched out our DNA, we have anywhere between 20 to 30,000 genes. That means in our DNA, we have essentially the recipe for how to make 20 to 30,000 different proteins. Something like the Ebola virus, which is a deadly hemorrhagic virus, it only has 7 genes, so it only has the ability to make 7 proteins. But Ebola, with its 7 genes and its minimal genetic content, is able to infect one of our cells and shut down our genetic material and then manipulate that cell into producing more Ebola viruses. So viruses don't have an abundance of genetic material, certainly not nearly as many genes as what we have, but the genetic material that they do have is quite important, and it can tell them how to infect a cell and then what to do once they get into the cell. So it can tell them how to shut off the cellular functions and then manipulate the cell into simply producing more viruses. Once a cell becomes infected with a virus, the only thing that cell knows how to do at that point is produce more viruses. So it didn't matter that it was a liver cell or a respiratory cell. It doesn't have those functions anymore. Once it gets taken over by a virus, its number one priority is just to make more viruses. There are other components that some viruses have and others don't. This is nothing that you would need to know for exam purposes, but some viruses need very specific enzymes that our cells would not have or the host cells would not have. So if that's the case, those viruses would have to make sure that they would take those enzymes into the host cell with them. So for example, the HIV virus needs an enzyme called reverse transcriptase, which is an enzyme that takes RNA and turns it into DNA, so it's the reverse of transcription. But our cells don't have this enzyme. So the HIV virus needs to make sure that when it infects a cell like a T cell, which it's known for infecting, that it carries that reverse transcriptase enzyme in with it because it knows it needs it, and once it gets into the human cell is not going to have it. So it has to make sure that it enters the host cell with it. So there are some viruses that require very specific enzymes, but for the context of this course, you need to know the components that are common among the majority of viruses, things like the spikes and the capsid and the nucleic acid core, and if we're talking about an envelope virus, then obviously an envelope. So the next part of this lesson is going through the animal virus replication cycle. So when a virus infects an animal cell, such as a human cell, what stages does it have to go through from the point of attachment to replicating more viruses to eventually leaving that host cell to go on and infect more host cells? There are 6 stages. The first being adsorption, which is basically when the virus attaches to a host cell. The second being penetration, which is when the virus enters a host cell. Uncoating, which is where the outer layers of the virus, like the capsid and potentially the envelope, are taken off of the virus to free the genetic material. Synthesis, where more viral parts are made, because obviously one virus goes in, but we're going to make hundreds of thousands to potentially even millions of more viruses. Assembly is when you put together all the parts of the virus to make mature viruses, which we often refer to as virons. And then ultimately release. The host cell is going to release all the new viruses so that those viruses can go bind to new host cells and the whole process can start all over again. So to be more specific and go through it in detail, the first step of a animal virus replication cycle is adsorption. So just kind of the overall gist of this is when the virus is going to attach to the host cell. So how this works is the virus has spikes that act as keys, and they are going to attach to receptors that act as locks on the host cell membrane. So right here we have, this is an envelope virus, so the spikes are going to be out there on the envelope, but the spikes on this virus are attaching to these purple receptors that would be on the surface of the host cell membrane. And if the spikes on the virus match the receptors on the host cell, then the virus is going to be able to progress to the next step, which would be penetration, which would be where the virus would enter the host cell. Now, this is a highly specific reaction. That's why I keep saying things like keys and locks. Not every virus can bind to every host cell. If that were the case, then we would be sick all the time. It's a very specific reaction where viruses have to be in the correct host, and they have to join with the right cell in a host to be able to go on to the next step, which is penetration. So viral attachment is highly specific. It's regulated through 2 things, one being host range and the second being viral specificity. So host range dictates what type of host a virus can infect. So there are some viruses that only target humans. There are some viruses that only target poultry. There are some viruses that only target dogs. So in order for a virus to establish an infection and attach to a host cell, it has to be in the right host. So for example, if I would become infected with COVID-19, the spikes on the COVID-19 virus are going to be able to match the receptors on my respiratory cells because that's a human pathogen. However, let's say my dog, because I'm infected with COVID, also gets exposed to COVID. So the COVID-19 virus gets into their respiratory system. The COVID-19 virus is going to try to attach its spikes to the receptors on my dog's respiratory cells. But since it's not in the right host, the spikes on the virus are not going to match the receptors on the respiratory cells of my dog. And for that reason, the virus is not going to be able to enter the dog's cells, and it's not going to be able to establish an infection. So in order for a virus to actually enter a cell, it has to get into the right host. A virus also has to, once it gets in the right host, it has to get to the right cell. It's not going to target every single cell. So for example, things like COVID, influenza would be targeting respiratory cells. Things like hepatitis would be targeting liver cells. So step 1 is kind of get in the right host, and then once the virus is in the right host, it has to find the right cell to match up with as well.